Hello everyone and welcome to our module on genetic principles. In this video I'm going to go through some vocabulary and some basic principles that you need to understand in order to watch the other videos on genetic diseases. So let's start with some basic terminology. The genome is the DNA contained in the nucleus of a eukaryotic cell. It's considered the hereditary material that can be passed on to successive generations of cells. Genes are portions of the DNA or genome inside a nucleus of a cell, and they code for proteins that carry out specific functions. A chromosome is a rod-shaped cellular organelle. You can see these under the microscope. I'll show you a picture of chromosomes in a minute. And a chromosome contains a single continuous DNA double helix strand, which contains a collection of our genes or DNA. And human cells have 46 chromosomes arranged in 23 pairs. The first 22 chromosomes are numbered 1 through 22, and then we have the sex chromosomes X and Y. And there are two copies of each chromosome 1 through 22. These are called a homologous pair. And a key point here that you need to understand in order to understand genetics is that in general we have two copies of most of our genes on a chromosome. This is a karyogram, which is a picture of the chromosomes inside a human cell, and you can see that there are 23 pairs as we just discussed. Most of the cells in the human body are somatic cells. These cells are referred to as diploid, meaning they contain two sets of chromosomes in 23 pairs like we just saw in the last picture. A small subset of our cells, however, are called gametes. These are reproductive cells, and they are haploid. They have only one set of chromosomes. They're designed to merge with another haploid cell to create a diploid offspring. The somatic cells in our body replicate through a process called mitosis. This involves an S phase of the cell cycle where the chromosomes are replicated into two sister chromatids. Then there is an M phase, which is where mitosis occurs, where the cell divides, and a daughter cell is created with copies of the chromosome. To understand this, I've drawn a picture at the bottom of the screen. So we begin with a somatic cell, which has a homologous chromosome pair, two copies of a given chromosome. For example, let's say this is chromosome number 11. In the S phase of the cell cycle, DNA is replicated and the chromosome becomes two sister chromatids, as shown in this picture here. The cell then divides to become two daughter cells, which contain copies of the original chromosomes. Reproductive cells undergo a process different from mitosis. It's called meiosis. This occurs in gametes. This generates cells that are haploid with one set of chromosomes. It's generated by the process of meiosis of cells called germline cells, and then two haploid cells will merge in fertilization to create a new diploid cells with half of the chromosomes coming from one parent and half from another. So a key point here, which is another thing that comes up again and again in genetics, is that in general offspring get one gene from their mother and one from their father. Some more terminology on this slide. An allele is an alternative form of a gene. It turns out that many genes have several forms. For example, the gene for eye color has multiple different forms, and that's why human beings have different eye colors. The same is true of many other genes. And in genetic textbooks and in genetic descriptions, this is often represented by a letter. For example, the A gene will have one form denoted by a capital A and another form denoted by a lowercase a. When we say a gene displays genetic polymorphism, we mean that it exists in multiple forms and has many different alleles. When we discuss the locus of a gene, the plural is loci, we're talking about the location of the allele on a chromosome. That's what the word locus means. So don't get confused. Remember that our cells contain DNA, which makes up our genes. Genes are found in multiple different forms called alleles. Each allele is located at a particular spot on a chromosome called a locus, and you can find the loci for various genes on particular chromosomes. The genotype is the genetic makeup of a cell or individual, and it often refers to the names of two copies of a gene. For example, if a person inherited gene A from their father and gene B from their mother, that person's genotype would be said to be AB. Or sometimes it refers to two alleles of gene A. For example, if gene A comes in the form big A and the little a form, the person's genotype might be two big A's, one big A and one little a, or two little a's. The phenotype, on the other hand, is the physical characteristics that result from a genotype. For example, genotype AB might equal phenotype blue eyes, and genotype BB might equal phenotype green eyes. The wild-type version of a gene or allele is the type that's common in most individuals. For example, for many genes that come in more than one allele, they will say the capital letter version of the gene, such as capital A, is the wild type. The mutant version of a gene or allele is different from the wild type. It's caused by a mutation, and for example, The lowercase letter often denotes the mutant in genetic examples, so an individual might have genotype capital AA, meaning they have two wild type or normal versions of the gene, or they might be a mixture with one capital A and one lowercase a, or they might have two copies of the mutant version of the allele. When we say a cell or an individual is homozygous, we mean that they have two identical copies of a gene. For example, imagine the A gene comes in two alleles, one we'll call capital A, the other we'll call lowercase a. A homozygous individual could have two copies of the capital A or two copies of the lower a version. A heterozygous individual or cell, on the other hand, has two different copies of the gene, such as capital A and little a. Mutations in our gene are what lead to different alleles and different versions of a particular gene. So let's talk about types of mutations. A germline mutation is a mutation that occurs in the DNA of sperm or egg cells, in other words, of gametes. This type of mutation can be transmitted to offspring, and when it is, the DNA mutation will be found in every cell in the body of that offspring that inherited that germline mutation. Somatic mutations, on the other hand, are acquired during the lifespan of a somatic cell. They are not transmitted to offspring. So, for example, if one of your skin cells acquires a mutation, that is never going to be passed on to your offspring. However, it will exist in all the daughter cells of that particular skin cell. A dominant version of a gene or a dominant allele is one that can determine the phenotype even among individuals who have only a single copy. It is often denoted with capital letters. For example, if a given gene has two alleles and one is denoted by capital A and the other by lowercase a, then individuals who have one copy of capital A and one lowercase a, or individuals who have two copies of capital A, will all have the A phenotype. In other words, the capital A allele dominates over the lowercase a allele. In contrast, a recessive gene or allele requires two copies to produce a phenotype. This is often denoted with lowercase letters. For example, only individuals who have two copies of the lowercase a allele will have the lowercase a phenotype. Individuals who have a capital A and lowercase a, or two capital A's, will have the capital A phenotype. Codominance occurs when there are different alleles for a given gene, but none are dominant over the other, and both alleles in an individual contribute to the phenotype. The classic example of codominance is the ABO blood groups. If you have the A gene, then your red cells will have the A antigen on their surface. If you have the B gene, then your red cells will have the B antigen on their surface. And if you have the O gene, your red cells will have neither A nor B. Individuals who are A and B express both the A and B antigens. That is because they are codominant. Neither one dominates over the other and excludes the effects of the other. The rare condition alpha-1 antitrypsin deficiency also displays codominance, and this is often tested on step 1. This condition may cause early COPD and liver disease, and I discussed the clinical aspects in detail in the video in the pulmonary section on obstructive lung disease. But for our purposes here, just know that mutations in the AAT gene, which produces alpha-1 antitrypsin deficiency, lead to this disorder. The M allele is the normal version of the AAT gene. If you have the S allele, you will produce moderately low levels of the alpha-1 antitrypsin protein. If you have the Z allele, you will produce severely reduced protein levels of alpha-1 antitrypsin. So the combination of alleles determines protein levels, and this is because they display codominance. So if an individual is MM, that individual will be normal. If an individual is ZZ, that individual will have a severe deficiency of alpha-1 antitrypsin. Other combinations lead to variable levels of disease depending on how much alpha-1 antitrypsin protein is produced. Penetrance is a genetic term used to describe the proportion of individuals with an allele that express the phenotype for that allele. And there are a number of diseases which display what's known as incomplete penetrance. This means that not all individuals with the disease mutation develop disease. It's often applied to autosomal dominant disorders where you would expect everyone that carries the gene to have disease. However, in disorders that have incomplete penetrance, not all patients with the autosomal dominant disease gene develop disease. And a classic example of genetic mutations that display incomplete penetrance are the BRCA1 and BRCA2 gene mutations. BRCA1 and BRCA2 are genetic mutations that lead to breast cancer. They are germline gene mutations and they are autosomal dominant. However, many women born with these mutations do not develop cancer, and this has important implications because although women that have these gene mutations are at increased risk, they may not develop breast cancer, and therefore if you are going to apply a cancer risk reduction strategy like prophylactic breast surgery, You don't know whether or not it'll benefit the patient since they may not develop cancer because these gene mutations display incomplete penetrance. Expressivity refers to variations in phenotype of an abnormal gene. This is different from penetrance, and this is often a point of confusion. To understand this, let's discuss a classic disorder that displays variable expressivity, and that is neurofibromatosis type 1. This is a neurocutaneous disorder where patients develop brain tumors and characteristic skin findings. It's an autosomal dominant disorder, so you expect all individuals to have disease, and in fact they do all have disease. This disorder has 100% penetrance. However, there's great variability in the disease severity, the number of tumors and the skin findings, and for this reason neurofibromatosis has variable expressivity, although it has 100% penetrance because everyone with the gene does get the disease. Pleiotropy is a term that refers to the fact that one gene or one gene mutation can have multiple phenotypic effects and traits. For example, a single gene mutation could affect the skin, the brains, and the eyes. And there are many examples of disorders which are caused by a single gene mutation, yet lead to findings in multiple different organ systems. For example, phenylketonuria or PKU can affect the skin. It leads to a characteristic body odor, and it also causes mental disability. Marfan syndrome leads to findings in the limbs, the eyes, and the blood vessels. Cystic fibrosis can involve the lungs and the pancreas, and osteogenesis imperfecta can involve the bones, eyes, and hearings. And there are many other examples of genes that have pleiotropic effects and genetic disorders that are caused by a single gene mutation but lead to abnormalities in multiple organ systems. For the next few slides, I'm going to discuss the two-hit origin of cancer. This refers to mutations in tumor suppressor genes that can lead to cancer. And the word tumor suppressor gene is very important because the two-hit origin of cancer only applies to mutations in these genes. Tumor suppressor genes are genes with many roles in cells. They are often gatekeepers that regulate cell cycle progression. Sometimes they are DNA repair genes. And if you have a heterozygous mutation for one of your two genes of a tumor suppressor gene, this will lead to no disease. You need to have mutations of both alleles to develop cancer. And for this reason, it's said that cancer requires two hits, meaning an abnormality in both copies of a tumor suppressor gene, or another way this is sometimes stated in genetic textbooks is that you need loss of heterozygosity in order to develop cancer related to a mutation of a tumor suppressor gene. Here's a picture from the web just to make this very clear. A normal individual has two copies of tumor suppressor genes, one found on each copy of a homologous chromosome pair. If they develop a mutation and lose one of the tumor suppressor genes, they still won't get cancer because the other gene will continue to function. This is said to be the first hit. However, if they develop a second hit and lose the other copy of the tumor suppressor gene, they will then have unregulated cell growth in that particular cell that has lost both copies of the tumor suppressor gene, and that will lead to no functional tumor suppressor effect, and that will lead to malignancy. The classic example of a tumor that illustrates the two-hit origin is retinoblastoma. This is the tumor in which the two-hit origin of cancer was originally described. It is a rare childhood eye malignancy, and 40% of cases are hereditary. In these cases, children inherit one mutated gene that's found in all cells of their body at birth. This is an example of a germline mutation. They then at a later point in childhood get a second somatic hit that takes out the second copy of the tumor suppressor gene. Because they are born with one abnormal form of the tumor suppressor gene, cancer requires only one somatic mutation in order to develop. Therefore, these children develop frequent and multiple tumors, and they develop tumors at a younger age. Contrast that with the sporadic form of retinoblastoma, which is non-familial. Children who develop this form of retinoblastoma need to have two somatic hits that take out both copies of their tumor suppressor genes, and two mutations in the same cell is a rare occurrence. Therefore, these children do not develop multiple tumors, they develop a single tumor, and in addition, the tumors usually occur at a later age. Another example of the two-hit origin of cancer is HNPCC or Lynch syndrome. HNPCC stands for Hereditary Non-Polyposis Colorectal Cancer Syndrome, and I discussed this in detail in the GI section, but for our purposes here, you should just know that this is an inherited colorectal cancer syndrome that involves a germline mutation in DNA mismatch repair genes, and these mismatch repair genes are another example of a tumor suppressor gene. So in this disorder, you have a parent who passes on two copies of a tumor suppressor gene to a child. One of those copies is abnormal because of a germline mutation. This means that gene will be abnormal in all the cells of the offspring's body. Eventually, the offspring will develop a second mutation in one of their cells, and this will lead to cancer. In addition, because that offspring has that mutation in all cells of their body, it will also be present in their germ cells, and therefore they can pass it on to the next family member, and therefore this tends to be a family which has lots of colorectal cancer, all because of this germline mutation. Another example that also involves the GI tract is familial adenomatous polyposis, or FAP. This is a germline mutation in the APC gene, another tumor suppressor gene. It always, 100% of the time, progresses to colon cancer. In fact, the risk of colon cancer is so high that people born with this germline mutation have a colon removal or a colectomy surgery prophylactically to prevent the development of colon cancer. And then one final example is the Li-Fraumeni syndrome. This is a syndrome of multiple malignancies that occur at an early age. It's sometimes called the SBLA cancer syndrome. SBLA stands for sarcoma, breast, leukemia, and adrenal gland. These are all places where tumors can develop. This is caused by another germline mutation, but this time in a tumor suppressor gene called TP53. This codes for tumor protein P53, which delays cell cycle progression to allow time for DNA repair. So once again, people inherit one copy of the TP53 gene that is abnormal. Over the course of their lives, in some cells in their body, the other copy becomes abnormal, and that leads to cancer. Now let's talk about mosaicism. This is a genetic term that refers to gene differences in cells of the same individual. So let's imagine that we have a whole bunch of blue cells which are replicating to create daughter cells, and one of them develops a mutation during the course of mitosis. This will lead to a new population of cells which has different genes compared to the other cells in that same individual. Therefore, this individual has a mixture or a mosaic of different genetic makeup, and therefore this is said to be an example of mosaicism. Mosaicism can involve germline or somatic cells. Germline mosaicism can be passed on to offspring. Let's imagine that we have a parent who has 3 different germline cells that are producing gametes to form offspring, and let's imagine that one of those germline cells develops a mutation. This is an example of germline mosaicism, and any gametes formed from this mutated cell will have a mutation that can be passed on to offspring. Pure germline mosaicism is very difficult to detect. It's not present in blood or tissue samples that are often used for genetic analysis. What this means is that offspring can develop disease that may appear to be a sporadic mutation, but in fact it's due to a germline mosaicism in the parent. So this can sometimes present in families as recurrent sporadic cases of disease in the offspring. What's really happening is that a parent has germline mosaicism that they are passing on to the offspring. In contrast, somatic mosaicism refers to gene differences in tissues or organs of an individual that do not involve the germline cells. For example, some cases of Turner syndrome involve mosaicism. Women with this version of Turner syndrome have some cells that are 45X and are missing an X chromosome, where other cells are normal and have 2 copies of the X chromosome. These tend to be milder variations of Turner syndrome. In addition, there are rare forms of Down syndrome that involve mosaicism. In babies born with these variants of Down syndrome, some cells have trisomy 21, which leads to Down syndrome, but other cells have 2 copies of chromosome 21, which is normal. There is a rare genetic disorder called McCune-Albright syndrome that is often used to illustrate the principle of mosaicism. This is a genetic disorder that affects many different systems. In particular, it affects many endocrine organs. One of its characteristic features is precocious puberty that can start at a very early age. For example, females may menstruate as early as 2 years old if they have McCune-Albright syndrome. In addition, there is fibrous growth that occurs in the bones. This leads to fractures and bony deformity. And then finally, there are characteristic skin findings. The skin becomes pigmented. These children can develop café au lait spots, and the skin pigmentation often has irregular borders that is classically described as appearing to look like the coast of Maine. Shown on the right side of the screen are 2 children with skin lesions from McCune-Albright syndrome. You can see how this child on the left has skin pigmentation with very irregular borders. And in case you don't know what the coast of Maine looks like, this is a map picture of Maine, and you can see how the coast is very irregular, just like this child's skin lesion on her chest. McCune-Albright syndrome is caused by a sporadic mutation that occurs during development of the child. It is not an inherited genetic abnormality. The mutation that occurs is a somatic mutation of the GNAS gene. This codes for the alpha subunit of a G3 protein that activates adenyl cyclase, and when this gene is abnormal, it leads to continued stimulation of cyclic AMP signaling more than is normal. The most important thing for you to remember about this very rare disorder for the Step 1 exam is what's shown on this slide. McCune-Albright syndrome is caused by a post-zygotic genetic mutation. It occurs after fertilization, and therefore only some tissues and organs are affected. In other words, it is said to display mosaicism, and the clinical phenotype will vary depending on which tissues are affected. So to understand this, just imagine that you have a developing fetus and it's got a number of different cells here, and one of them develops this mutation. All the daughter cells of this mutated cell will have the McCune-Albright syndrome genetic mutation, but the daughter cells of these other cells will not have the mutation, and therefore the child will develop as a mosaic of genetically modified cells, some being normal, some having the mutation. If there happens to be a germline mutation that leads to the McCune-Albright genetic mutation in all of the cells, that is a lethal condition. In that case, the entire body is affected. So in other words, cells with this mutation can only survive if they are mixed with normal cells in the individual. What this means is if you see a child in clinical practice with McCune-Albright syndrome, that child must have mosaicism. If the child did not, they would have died and they would not have survived to come in for clinical evaluation. I'll finish this video with a couple more terms that you should be familiar with. The first one is genetic heterogeneity. This refers to when the same phenotype develops from different genes or mutations. For example, different mutations of the same allele can lead to the same disease. In other words, there is not a single mutation that leads to the disease. Multiple different types of mutations can cause the disease. We'll talk more about that in a minute. A second example is when different genetic mutations at different loci on the chromosomes cause the same disease. This also is an example of genetic heterogeneity. So the point here is that for many genetic diseases, multiple gene mutations often cause the same disease. Or another way to look at it is that many clinical diseases have multiple genotypes that lead to those disorders, and this is referred to as genetic heterogeneity. One subtype of genetic heterogeneity is allelic heterogeneity. So by way of review, an allele is an alternative form of a gene. So imagine that allele 1 is formed by mutation X of a particular gene, and allele 2 is formed by mutation Y of that same gene. But both X and Y cause the same disease, and X and Y are found at the same chromosomal location or position, therefore they involve mutations of the same gene. This is termed allelic heterogeneity. It means different alleles or different mutations lead to the same disease. And the point here is that many alleles possess multiple mutant forms that all lead to the same clinical presentation or clinical disorder. So I've summarized this at the bottom. You have one disease caused by multiple genes, but all those genes are variants of a gene at the same location, therefore they are allelic variants, and therefore this is termed allelic heterogeneity. A couple examples of allelic heterogeneity are shown on this slide. Beta thalassemia, which I discuss in the hematology section, involves mutations of the beta globin gene, and there's a wide spectrum of disease severity depending on the mutation. People that are said to possess the beta 0 form of the gene have no function. People who possess the beta 1 form have some function, and I talk about this in more detail in the hematology section. Cystic fibrosis is another example of a disease that displays allelic heterogeneity. Cystic fibrosis is caused from mutations of the CFTR gene, and there are over 1,400 different mutations that have been described that can lead to various forms of cystic fibrosis. Another subtype of genetic heterogeneity is locus heterogeneity. This refers to when mutations in different loci on chromosomes cause the same phenotype. So imagine that we have a chromosome, say it's chromosome number 1 here, and a mutation at this gene right here causes a disease, and then over here we have chromosome number 3, and a mutation here causes the same disease. This is the same disease coming from genetic mutations in 2 different loci on chromosomes, and this is an example of locus heterogeneity. And one example of this in clinical practice is retinitis pigmentosa. This is a rare disorder that causes visual impairment, and there are many different forms of this disorder. There's an autosomal dominant form, a recessive form, an X-linked form. It turns out that mutations at 43 different loci on chromosomes can all lead to this disease. Therefore, retinitis pigmentosa is said to display locus heterogeneity. And I've summarized the key points at the bottom. In locus heterogeneity, we have one disease caused by multiple genes at multiple different locations on chromosomes. And that concludes our video on genetic principles.